Um, Great. Thank you guys for joining in. My name is Manny Azizi. I'm from the University of California, Irvine. And this workshop is going to be largely about in-series elasticity, which in and of itself, if you focus just on the tissue, can be a little bit dry and boring, but it'll be really in the context of trying to understand implications for muscle mechanics and whole animal or whole person energetics. So that's where we're headed. What is in-series elasticity? So I'm going to start with some really basic stuff first, just to give context to our trainees before diving into some more complicated stuff. In-series elasticity is really considered. Do you guys see that the share screen on my screen right now? Should I move it? The little thing that says stop share?
Uh,
No?
no, we
Okay,
just slides.
great. In-series elasticity exists in both invertebrates and vertebrates. I consider this really a ubiquitous part of the animal kingdom. I think there's some elasticity and elastic tissues in nearly all organisms. In invertebrates, which I'm not going to focus on much today, you can think of things like the apodeme, modified exoskeleton structures as well, like the semilunar process or the exoskeleton itself, are really good elastic structures that are capable of deforming, storing mechanical energy temporarily, and releasing that. And in fact, some of the most resilient materials come from the invertebrate world, where the resilience of that material can be quite high. In vertebrates, mostly what we talk about when we talk about in-series elasticity are tendons, which we know quite a bit about, and aponeuroses, which I also consider to be sort of tendons, except that they're more like broad sheet-like tendons that allow for muscle fibers to attach to a broader insertion site than a long free tendon, for example. And we've done some work on aponeuroses in the past, showing the complexity of how they're loaded relative to free tendons. But for today, I'm going to sort of focus primarily on tendons. Tendons, as most of you know, are made primarily of collagen. And so when we look at this hierarchical structure of the tendon, we can see that tropocollagen is one of the primary proteins, and the helices that form are collagen fibers in that 10 to 50 micron range. That then leads into higher levels of organization like tendon fascicles, which is a level of organization where we've done some work where you can isolate tendon fascicles and pull on them individually. And then you get to the whole tendon, which is made up of many fascicles. Interesting things about tendons is that they're fairly acellular. There are tenocytes and fibroblasts in place that are laying down and repairing collagen, and there's some vasculature, but the general metabolic activity of tendons is pretty low. And that can result in a lot of downstream consequences, particularly when you think about the fact that when you're operating in series with a muscle, the properties of the tendon and the properties of the muscle need to be well tuned. And if remodeling rates differ significantly between these tissues, you can start to have mismatch as a result of things like weightlifting or bed rest, anything that changes muscle properties rapidly. Those changes don't really show up in tendons for a long time. And so you can have these periods of mismatch. Now, what we do frequently in my lab is simply just grab a piece of tendon or grab a tendon fascicle, pull on it, and measure the deformation and the force that occurs. Tendons we can think of as nonlinear in their behavior, in that unlike what would be considered a true spring or a Hookean spring, they really have two regions. They have this toe region down here where it's relatively compliant, leading into a more linear region here where we can characterize the linear stiffness of that material. This nonlinearity in the toe region really results from the fact that the collagen fibers that are in tendon are sitting in a crimped position at a low length. And so you can see here this sort of crimping looks like bad hairdo from the 80s, that really results in some compliance in the very low strain regime of tendons, where that initial pull is actually straightening out those crimps rather than directly pulling on the material itself. So we have some nonlinearity that's interesting, and then we can normalize this so that we can compare across sizes or different systems. If you normalize force by cross-sectional area and normalize the length change as a proportional length change, we can then get to something that's much more comparable across systems, which is the elastic modulus rather than the stiffness itself. Now, most often tendons aren't just stretched, they are cycled through length changes. And so if we take a tendon and we drive it through some sinusoidal length change, what we notice is that it does return a good portion of that energy back to us. So as we load energy into the system and then bring that system back, what we see is that we've got this area under that loop, that's the energy lost in that cycle. That really defines the resilience of a material, the amount of energy lost as you cycle through it. And for tendons, that happens to be somewhere between 80% on the low end, that's the energy we're getting back, to almost 90% on the high end, which would be so we'd be getting back 90% of the energy that we would load into a tendon. However, the fact that it does lose energy with every cycle suggests that this isn't really a spring, that it's really more of a viscoelastic tissue, and that we should really start thinking about it that way. And the work loop or the hysteresis is not the only source of that viscoelastic behavior. If we take a tendon, for example, and we just pull it and then hold it at that longer length, what we see is that the force goes up, but it doesn't stay up here. We have this creep and loss in force, which is the stress relaxation behavior of tendons, which is also a characteristic feature of viscoelastic materials. So really, we're thinking about this as, yes, it's a pretty good spring, but it's really a viscoelastic material. And we have IMSI projects that are ongoing, working with Kisa and Miles Valencia to start to use system ID to characterize the spring stiffness and the damping coefficient of the material, and to start to think of it more as a material that's viscoelastic. Okay, that's tendon in isolation. I want to move past that because really what we care about is how are these tissues used in movement. And what now doesn't seem like that long ago to me, but it's been a while. Tom Roberts and I came up with a general framework for understanding the function of tendons, and we really borrowed from a 2000 paper by Michael Dickinson and the Integrative Biology Biomechanics group at Berkeley that really started to define muscle function as being varied and really being defined as a behavior or behaviors where the muscle acts as a motor and produces mechanical energy, as a strut where it just produces force, or as a brake where it's actually dissipating energy with every contraction. And so we really thought about tendons in that same way to try to understand, well, what are tendons likely to do in these various muscle function scenarios? And so we really thought about three ways in which tendons can operate during locomotion. First being power conservation, the second being power amplification, and the third being power attenuation. Power conservation is the one that's most well known and was when we wrote this paper. And so we'll start there, although I won't spend much time on that today. With power conservation, what we're really thinking about are behaviors that are cyclical, where the mechanical energy of the center of mass from the previous stride can be transferred to the subsequent stride. And the way to do that is to cycle that energy through the tendon. And so if we look at this schematic that I have there, the red indicates where there is a high mechanical energy. And so what we see in power conservation is that the center of mass has high kinetic and potential energy. It's in the air. It's about to come down. And when the foot hits the ground, for example, and the ankle flexes, the mechanical energy from the center of mass gets loaded into our tendon, while the muscle is simply producing force and operating isometrically. So this would be a scenario where the muscle is operating as a strut, but the tendon is really the one that's cycling the energy from the center of mass with each subsequent stride, allowing us to recycle the energy from a previous cycle into the next cycle. So in this case, we have energy flowing from the body to the tendon, and then as that tendon recoils, it helps accelerate the body once again. So in this case, the muscle is really isometric. And so this is where the muscle is behaving like a strut, but much of that strain energy is being cycled directly through the tendon. So behaviors like walking, running, and hopping are great examples where the tendon is helping us minimize the metabolic cost of locomotion by allowing muscles to minimize the amount of mechanical work that they're doing. Okay, I will say, anytime anybody has a question, you can raise your hand. I'll try to keep an eye on the chat as well. But if Victoria can keep an eye on the chat and transfer questions, I would be greatly appreciative of that. Okay, next I want to talk about power amplification, which is one where the body is actually starting from a standstill. So you can think of this as behaviors where we really want to increase the mechanical energy of our center of mass. So let's say we're starting from a standstill and we're accelerating. So behaviors like jumping, where we're low and we want to get higher. And so that's obviously an increase in mechanical energy. Accelerating or running up a hill, all those are behaviors that require our muscles to impart additional energy onto our center of mass. In these scenarios, we can amplify the mechanical power of our muscles through the utilization of in-series elasticity. And in this case, as we're looking at the energy flow, what we really have is from a standstill, we have the muscle firing. The muscle is producing mechanical energy, which is being transferred directly to the tendon in this case. Now, an important step or requirement here is that we need something to resist the tendency for that ankle or that joint to extend as that muscle is producing force. So these behaviors require something like a latch that at one point resists the forces that the muscle are producing, allowing time for that energy to be stored in the tendon. Once that latch is released, the energy from that tendon is released, allowing the center of mass to get accelerated. Okay, so behaviors like jumping, accelerating, or incline running really rely on a combination of muscle work being transferred to the tendon and then being released onto the center of mass. The key here is muscles are going to be much more power limited than tendons are. And so the benefit here is really that by having a latch in place, our muscle can contract much more slowly and economically and generate much higher forces than would be required to actually directly accelerate this mass. So the muscle can perform at a much slower and more optimal place, allowing that energy to be stored in the tendon, which gets released much more rapidly. So where we get the power amplification is that the amount of mechanical work is the same because it came from the muscle, but the duration over which that mechanical work is released is much shorter in a tendon than it would be for a muscle. So we're really playing with time constants here as we're increasing the power that's associated with the behavior. So in this case, what we really have is a flow of mechanical energy going from the muscle, being loaded into the tendon, and then being rapidly released to accelerate the body. Okay, final one I'm going to talk about is power attenuation, which occurs during behaviors where the muscle is aiming to dissipate mechanical energy, things like landing from a jump. So the back half of that jump is you're going to come down and hit the ground. Or decelerating or running downhill. All those are behaviors where the center of mass has high amounts of mechanical energy that we want to get rid of. We want to slow the system down, and our muscles really are good at that by stretching actively and really converting mechanical energy into heat and dissipating it as that. So in power attenuation, what we really get is a flow of mechanical energy that goes from the body. So the mechanical state of the body is quite high to start with. as the person, let's say, lands, that energy goes into the tendon before the tendon actually does work back on the muscle. You may think, okay, well, why can't the muscle do that directly? It can. The benefits of utilizing tendons for power attenuation is that they minimize the direct high velocity or high power input to the muscle and are much more controlled in the way that the tendon releases energy back onto the muscle. So it's a much more predictable way to load the muscle compared to directly loading it with the environment. So we really, in this case, have the tendon acting as this buffer between the energy that's coming from the environment or the center of mass and the muscle that's ultimately having to be stretched in order to dissipate that energy. Okay, those are going to be the three things I'm going to talk about. And so let me start by talking a bit about power conservation. And I'm going to use some classic work to sort of make this point. In 1992, Russ Bodinett made a really interesting observation, which is that if you track the rate of oxygen uptake or rate of oxygen consumption in a locomoting animal, and if you have, let's say, a 5 kilogram animal that's running, we know that as the speed increases, the metabolic cost of that behavior increases linearly across most animals. That's simply because as our speed increases, our stance time gets shorter and shorter. We still have to support our body weight with every stride. And as a result, we're either having to utilize more expensive muscle fibers or have our fibers contract more rapidly to accommodate that shorter stance period. That's a well-established pattern that had existed in the literature. What was found with wallaby hopping was that, in fact, once they start hopping around 3 meters per second, they transition from a weird quadrupedal running gait to a hopping gait. Once they transition to that hopping gait, their gait or their metabolic expenditure is largely independent of speed. Okay. And so regardless of how short that stance time is, it appears that that doesn't bear a significant cost in terms of metabolic expenditure. Following that observation, Russ worked with Andy Bewinner to actually make muscle measurements in wallabies. And here we have force in the top. We've got length of the muscle fascicles in the middle and EMG activity shown there. What I want you to take away from this is that, yes, during stance, there's this large period of force. However, there is very little length change in that muscle. And so what was found is that, okay, the muscles really aren't doing much mechanical work at all. During the stance phase of locomotion, they're operating isometrically. And we know that the ankle in this case is undergoing significant flexion and extension as the animal is hopping along. And so we know that there's length change going on somewhere. It's just not happening in the muscles. So where it's really happening is that that length change is being cycled through the tendon in order to, or during the stance phase where force is being produced. And the muscles are really producing force isometrically. So what they mapped in that paper was that if you track the amount of elastic energy that's being stored versus the amount of muscle work that's being done, what we see is that as the animal is increasing speed, it is not increasing the amount of mechanical work it's doing. It's increasing the amount of elastic strain energy that's being cycled through the tendon. That is metabolically inexpensive. It's largely free, right? Rather than the work that muscles are doing. So this flat pattern of muscle work across that speed range really helped explain this pattern where, or this independence, the speed independence of metabolic expenditure. So just to schematize it, what they suggested was as the foot hits the ground, as the ankle flexes, the muscle is producing force isometrically, the tendon is being stretched. In the second half, as the ankle is extending, that tendon is releasing that energy and accelerating the animal forward. Now, This has some important implications for metabolic expenditure, but it doesn't get quite at the mechanism of what is allowing that savings to occur. There's really two alternate hypotheses here. One being that doing mechanical work is simply more expensive than just producing force. That's a work-based metabolic hypothesis. Um, the alternate hypothesis is one that focuses more on force and the cost that is required to generate the amount of force needed to support your body weight. To sort of unravel this pattern, Natalie Holt had this really excellent paper in 2014 where she did some really painstaking work to not just vary the pattern of length change in the muscle and measure its force, but to also measure the heat that it's producing, which is a direct measure of metabolic expenditure at the muscle level. And so she was really measuring the cost of a muscle contraction when in A, the muscle is isometric, in B, it's undergoing a stretch shorten cycle, in C, it's just shortening, and in D, it's really being hit when it's lengthening. So she varied that strain pattern in order to determine, well, does it actually cost more when a muscle produces mechanical work versus an isometric contraction? What she found was that no, in fact, it doesn't. When she plots energy for three conditions, the isometric, the stretch shorten cycle, or the one where it's undergoing shortening, what she showed is that in fact, the isometric contraction costs just as much. It's largely indistinguishable from a stretch shorten cycle or even direct shortening. So the idea that muscles doing work cost more than muscles producing force, I think doesn't quite hold up. And so this really supports the cost of force hypothesis, which simply suggests that yes, there is a benefit to being isometric, but that's largely because as you start to shorten with some reasonable speed, you are having a reduction in your force capacity to some degree. And that reduction in force capacity will then require you, if you're trying to match the same level of force, to start to recruit a larger volume of your muscles in order to meet that force requirement. So this seems pretty reasonable to me. I'm certainly in the cost of force camp when it comes to the metabolic cost of locomotion, though I realize that deformations associated with sliding filaments likely have some small amount of cost associated with them as well. So the observations of maintaining an isometric behavior have their benefit regardless of what model of the metabolic cost of locomotion you believe in. If tendons allow muscles to stay isometric, they do bear a benefit. So during power conservation, we have some metabolic savings that are associated with the recycling of metabolic energy from one cycle to the next. Kisa, go ahead, please.
Yeah, Manny, so I just had a question about
Sure.
the previous slide and that paper by that beautiful paper by Natalie. And yeah, so, and I guess what I just wanted to say is that, there's a lot of evidence that when you stretch a muscle, like in a SSC, and there's human work and other work on this, before it shortens, like you can actually see this energy, and this is at the muscle level without any tendons, is substantially lower. So I just wanted to say, I think like that this is evidence for energy storage also in the muscles. And yeah, that was my main point, is that I think um, that we've kind of systematically underestimated how much energy like the actual muscles are storing, particularly when tendons aren't super stiff.
Yeah, I agree.
Okay.
Okay. Next, I'm going to focus on power amplification. This is an area where we did a significant amount of work, particularly in collaboration with a large interdisciplinary team. We worked for many years on various aspects of this in lots of different systems. So I'm just going to really sort of skim the very top of this. In 2019, we had a paper by Mark Ilton, where we really developed an extremely simple model of the components that are required um, to utilize this kind of power amplification. We came up with four components, a four-component toy model. We have the motor in red, we have the spring in blue, we have a latch, and then we have a projectile. So what we have is the motor producing mechanical energy. In this case, it's going to shorten and it's going to push our projectile down and it's going to store energy in this spring. This latch holds the position of our loaded system. Once the latch moves out of the way, that allows this spring to then accelerate this projectile and send it into the air. Okay. This is exactly the same as a bow and arrow or a slingshot. This is really utilizing a combination of motors, springs, and projectiles with a latching mechanism in place in order to increase or amplify the output of that system. So you can imagine that in a bow and arrow, for example, your arm is the motor, the bow itself is the spring. You're loading energy into that spring. Your fingers holding the arrow are really the latch. When they let go, you've unlatched the system, and that arrow is going to fly, and it's going to fly much further than you can throw it, right? So this increases the output of let's say the power output. You can think of it as kinetic energy at takeoff, the height that that projectile goes to. It doesn't really matter what performance parameter it is. It's going to generally do better than the muscle. So we sort of thought about it this way. During the loading phase, we're loading potential energy into our spring. It's going to be latched, and so we're going to have that energy sort of stay up here. There's going to be an unlatching phase, which is the rate at which that yellow box gets out of the way. And then once it gets out of the way, we see that there's some rapid extension of that spring that then sends our projectile up into the air. What's key in these systems are really a couple of things. One is how good that latch is. Okay, and we played around with making that box a rounded edge, lots of and changing the speed at which you pull it back and so on. The shorter that unlatching phase is, the higher the performance of the system. Anytime that slows down, you're actually losing energy to the latch itself, because during the unlatching, you're starting to lose some of the energy here. Okay, so the longer it takes, the poorer the latch, the more of that energy gets sapped from the system before you're actually applying it to the projectile that you're accelerating. So that's one thing. Latching's extremely important, and it's actually really regulating the flow of mechanical energy. So it's really the critical piece in this puzzle. The second is, if you start to play around with the mechanical properties of the motor and the spring, their tuned relationship is also critical. That in fact, if those are not really working particularly well together, you're simply not going to have the system perform as well. So taking this model, we really thought, okay, well, we got to have to replace that generic motor with behaviors that are more muscle-like. And so we know that the force length properties of skeletal muscle really restrict deformation or limit the amount of length changes that are available to the system. When they do, that's likely to influence the properties of the spring that make sense. So in this case, what we were able to show was that, in fact, if you start from some long length, but your spring is really compliant, rather than load a lot of energy into that system. And so what I'm doing here is I'm starting a spring and then I'm ending it here, right? So as the muscle contracts against the spring, The spring's very compliant. The muscle shortens a lot quickly, but because there isn't a lot of force associated with that spring, there isn't a lot of energy being stored in it. So when it's too compliant, we get to lengths where muscles aren't producing great force before we've been able to load a lot of energy into the system. Okay. If we're too stiff, there's very little shortening that's occurring in the tendon, so the muscle effectively is more isometric with little shortening of the tendon itself, and so the area under that curve is also a little bit small. Those are both cases where we're able to calculate what actually the optimal is, which is right in between those, where the optimal is not too stiff, not too compliant, allows a significant amount of shortening, but doesn't shorten so much to where the muscle's forces start to really drop off. And so the biggest area we get, the area under that curve, is really the energy that's stored in the tendon. And so the size of that triangle should be interpreted as how much energy I was able to put into that tendon. So this matching between tendon and muscle properties really starts to be critical. We thought we could test this in a system that I've worked on for a while, which are jumping frogs. And in this jumping frog, we're going to see a jump off this force plate. In this case, the tendons are the springs, the hind limb muscles are the motors. The latch in this case is actually the mechanical advantage of the limb itself, which changes dynamically throughout the jump. So when the animal is in this posture where the limb is highly flexed, the mechanical advantage is quite low. And as the muscle produces force, that low mechanical advantage combined with the inertia of the body itself hold the animal long enough for it to store energy in its tendons before it starts to extend its limbs, creating a positive feedback loop where the increase in its mechanical advantage starts to release the energy in the tendon. Here's the animal taking off, and you can see that it's quite an excellent jumper, accelerating nicely out of our view. We thought the idea of matching muscle and tendon properties could potentially help us with a conundrum that we had when we compared 3 species of frogs and showed that their behavior during jumps are significantly different. So these are power in watts per kilogram of muscle mass plotted against time for a jumping tree frog, a bullfrog, and a toad. So we see that the tree frog generates significantly higher power outputs during a jump compared to the bullfrog, and the toad comes in dead last. Okay. Now, our original foray was to try to explain this difference through the capacity of their muscles. This was in 2011. But it turns out that despite the fact that the jump power increases substantially across these 3 species, their muscle power, shown in white, is largely constant across all of those. So differences in mechanical power capacity of skeletal muscles were not really able to explain the differences that we observe across these 3 species in their jump performance. What we were able to show once we started to really dig into the mechanical properties of the tendons and muscles is that what we found was that if you track the behavior of the muscle and the tendon, and so in this case, I'm showing you a sample where the muscle's starting here, shortening to some length, producing force, and the area gives us the capacity for that muscle to store mechanical energy. Kisa, go ahead.
Finish your slide and then I'll chime in.
Okay. So what we see is that the area defines the amount of mechanical energy that each of those muscles can store in their tendons. What we see is that a couple of things differ when it comes to the tree frogs, potentially explaining their high performance. One, the specific force or their stress capacity is quite a bit higher, particularly if you normalize it to muscle mass. They have highly pinnate muscles that are capable of higher forces per muscle mass and are able to get to these higher forces than the other species. Matching that high force capacity is the fact that their tendons are also stiffer. And the way you can interpret that is, what does the slope look like in that line when the muscle is shortening against the stretch of tendon? Really, the slope of these lines is also the stiffness of the tendon. So here what we see is that tree frogs have evolved higher force capacity through changes in the structural properties of their muscles and are operating with stiffer tendons in order to increase the amount of mechanical energy that they can store in their tendons during a jump. So this mismatch, this idea that the tendons and muscles have to match really does provide some level of explanation for the variation in jumping performance that we see across three species. Kisa?
Yeah, so I guess my question is, how did you estimate the peak power of the muscles? And, was that like the isotonic peak power?
It is.
Yeah, yeah. So like my little chirpy sort of comment would be, so, that might or might not differ, if we did like if our group can think of a measurement of how we might estimate the dynamic muscle power when you actually have changing load. It might be the same still across the different species, but it might just be that the isotonic constraint is maybe systematically underestimating what the muscle power actually is.
Well, it has to be sustained though, because this is a behavior that takes, let's say, 300 milliseconds of loading. And so if you think about the muscle power requirements, they can't be transients. They can't be transient measures of muscle power. They have to be sustained measures of muscle power.
Right. I would just say like I was thinking of like if you look, for example, at the recent paper, from Peterson and Roberts, that there is some elastic recoil of the muscles as well. And um, so I think that, um,
But the
that for
elastic
a
recoil
given,
of muscles goes in the wrong direction.
well, I think for a given, I think for what my interpretation is that, and we can talk about more about this offline, but for a given velocity, muscles can actually produce quite a bit more force when their load is reducing than they can when the load is constant.
I agree.
Yes. Yeah.
Okay. The other the other bit of information that we were able to glean from that little toy model that we came up with was that in fact there's a really interesting scaling relationship between when these tendon-driven behaviors make sense and are useful and are beneficial in terms of performance, and when really just directly actuating the system with muscles is more beneficial. And the way this was derived was for a very specific set of scenarios where we gave some muscle-like properties to our motor. And we ran sort of two different spring models, one where it's an ideal spring and one where it's got some mass and some realistic properties. What we found was if you measure, let's say, maximum power, we also did this for maximum kinetic energy of the projectile, maximum momentum. So we had lots of different metrics that we looked at. The pattern was quite consistent, although the mass was a little different based on what metric you use, not significantly though on a large scale. What we found was that if you are small, springs make a ton of sense, and that your power outputs being spring driven significantly outperform muscle-driven behavior where you're directly actuating the system with skeletal muscles. And then you get to some transitional length or size, where beyond that point, you start to really lose the benefit of rapid delivery of mechanical power from a tendon that exceeds the rate at which power can be produced by the skeletal muscle. That happens to be on the order of, let's say, 5 to 10 gram masses. And the way that the reasoning behind that is that as you get bigger, that the mass is going to have inertia associated with it. It's going to take time to accelerate that mass. That time effectively saps that power that the tendon is producing. So it doesn't matter how fast the tendon is recoiling. If the mass is so high that its inertia is going to take some amount of time to get to its peak velocity anyway. So in this case, the higher body masses tend to have this higher inertial constraint, which then limits their utility, the utility of spring-driven systems. What was interesting was I was on this team, my system was frogs, and so we were really right in that transitional zone where for some species, it made a lot of sense to be spring-driven, and for some species, it made a lot of sense to be directly muscle-driven. The way we started to think about this was that, well, maybe these things at some point, these things may not be mutually exclusive, that you may not simply be a spring-driven system or a muscle-driven system, but that those things may be able to work in concert once the system gets slow enough for the muscle to be able to generate the powers needed to continue to contribute to the takeoff phase. And so we came up with this prediction that, well, I think frogs are in this nice little range where they can simultaneously utilize both spring and muscle actuation during their takeoff phase. The way we tested this prediction was through what we call virtual reality muscle experiments. In these experiments, what we effectively have is an isolated muscle tendon unit where we're independently measuring the length changes of the muscle fascicles. In turn, that gives us the length changes of the tendon. We're also stimulating this muscle artificially. However, rather than just measuring the force output of various contractions, what we have is a real-time feedback controller that's in place that allows us to take the force output of our muscle in every time step, feed it into an in silico model of a jointed system where we've got a mass up on top. This is really not there. That's actually the muscle tendon unit in real life that's producing the forces. And it's actuating it through this jointed system that's got some leverage that we matched to our frogs. It's got a mass that we matched to our frogs. And so we're really doing these sort of hybrid experiments where you have real biological tissues interacting in real time with a model. Okay, so this is a vertical jumping model. It's a pretty simple thing that we could code up and have the muscle interact with it in real time. And then in order to really test the relative contributions of muscles and springs in this system, we did this one additional manipulation, which is that we altered the temperature of our system. That allows us to alter our muscle properties significantly while retaining our tendon properties. So tendons are not really temperature sensitive. They're not undergoing enzymatic reactions. They're a mechanical tissue, whereas muscles are undergoing enzymatic reactions. And so as a result, they're going to be much more temperature sensitive. So we ran this model in these experiments. And so same with the frog system, the mechanical advantage of that jointed model is increasing through time. It's storing energy, it's releasing energy, it's behaving in ways that we think resemble our frogs pretty well. So here we've got the joint angle of the model at 10 and 20 degrees. Those sort of get to the same place. They all take off. regardless of the temperature. It just takes a while longer for the 10 degree to do that. Here we have muscle force. We have higher forces in red. That's not really a thermal effect. That's more of an effect of the rate of force development increasing significantly with our increase in temperature. And then we've got our fascicle lengths. That's our muscle shortening during this behavior in order to store energy into the tendon and then release it when the force starts to come down. The part of this trace that we were really interested in were these parts down here, where you have significant amount of shortening really near the end, like really close to the takeoff of this muscle, of this model. Here's what we were able to find. So I have force length curves plotted here. I've got two conditions, cold in blue, hot in red. We have our muscle shortening. That dashed line is kind of the inertial constraint of our model. We have to get to that force before the model starts to accelerate. What we see is that when it's hot, muscle develops force, shortens to stretch the tendon, but it overshoots that threshold because its rate of force development is pretty high. Stores a good amount of energy in that first phase, which is pre-threshold. Then it overshoots the threshold in phase 2, and then it continues to shorten during phase 3, which is when the tendon is also recoiling. So in the first part, we have the muscle loading the tendon. Actually, in the first two parts, we have the muscle loading the tendon. And in phase 3, the tendon is unloading, but the muscle is also continuing to shorten rapidly and produce high power outputs. So what we really have is evidence of a system where the muscle is storing energy into the tendon begins to recoil, but the muscle is not done and is not getting out of the way. It can continue to impart mechanical energy into the system during the takeoff phase, okay? So what we see when we compare the two temperatures is that as you slow the rate of force development down, we don't overshoot our threshold by much when the muscle gets cold. But interestingly, where we see the biggest difference is in this third phase where the muscle is imparting additional power onto the system. We see that when we decrease the temperature and we decrease the capacity of the muscle to generate mechanical power, that's the part where we start to lose. Now, what this helped explain was something interesting, which was that it's been long held that elastically driven behaviors are not really going to be temperature sensitive, but also that frog jumping is highly temperature sensitive. And so that mismatch in ideas is really explained by the fact that this isn't a purely elastic behavior driven by springs, but it's that it's really a combination. And that the part you start to lose when the temperature gets cold is that additional muscle power during the unloading phase of the behavior. I can show that more quantitatively here. What we see is that the amount of energy stored slowly during that early phase is the same across temperature. The overshoot helps us load a little bit more energy when things get hotter. But interestingly, the power of the recoil of the tendon and the power of the muscle during unloading is where we see the highest temperature effects. And in fact, what we see is that there's almost a 50-50 split between the amount of energy that's coming out of the tendon and the amount of additional energy that's being put back into the system by the muscle during the unloading phase of the behavior. So really, what I take away from this is that the utilization of elastic energy doesn't have to be perfect. It doesn't have to be discrete where you describe a system either as utilizing spring actuation or utilizing direct muscle actuation, and that those two things can coexist. And to me, that really broadens the realm of the utilization of elastic energy because it provides the opportunity to use it alongside muscle-driven behavior. Okay,
Can I ask a quick question here?
sure.
The temperatures here, the cold and the hot in these simulations,
Yeah.
um, is that like an average across the overall system? Like where is the temperature actually?
Yeah, so this is the bath that the muscle tendon unit is sitting
Mm,
in is literally at that temperature.
okay.
So our model is agnostic to the temperature, but our muscle
Mm,
tendon unit is being affected by varying the temperature of the system. So we're really varying the muscle dynamics directly here.
okay, that makes sense. And is that because it's in a bath, I assume the temperature stays pretty constant throughout the
Yes.
whole system?
It's a circulating bath set at a constant temperature. Yeah.
In vivo, then, does the temperature of the whole system kind of stay pretty constant there as well, or does it kind of vary throughout as you go deeper into the body and things like that?
It varies. I mean, these are ectothermic animals, so they're
Mhm.
not, so they're at the will of their environment. However, I will give this nice anecdote, which is that when we went to the Calaveras County frog jumping competition,
That sounds
almost
fun.
all of the best competitors had this secret bath that the
Mhm.
frogs were in just before the competition. And to me, that is just heating up the frog up to a certain temperature in order to maximize its power output.
Amazing.
But interestingly, really, we're able to explain why there is a temperature effect in a system that we've long thought was elastically driven.
Mm.
Jill?
I have a quick question. So I'm really curious, is there a latching mechanism that can be applied in this scenario?
Yeah, so the latch here is the same as the latch in the frog, which is that the we're starting our model in a highly crouched posture
Yeah,
that is effectively operating with a really low mechanical advantage. And then the mass has sort of an inertial constraint. That's kind of that dotted line that you have to overcome that inertia in order for the mass to start to accelerate.
where I'm going with my question is I'm thinking about skilled jumpers in technical situations where they can manipulate that.
Yes.
Yes, so this is very, very interesting.
Um,
Yeah.
The most interesting thing we've done with this system is to jump these animals off of compliant substrates, where
Ah,
you're effectively you're adding a spring in series, an additional spring in series, effectively by having them jump off a diving board effectively. And what's interesting is I don't think they're consciously doing this, but their behavior slows down because
yeah.
they're able to utilize more of the recoil of the substrate simply because their latching mechanism works less well when the ground gives under them and forces their limb to extend prematurely.
This is something that's highly regulated in divers, so this
Yeah,
is very interesting
yeah,
because you want to get the most out at the right time.
exactly. The
Yeah,
timing of that. So jumping straight, yeah, you can see divers like hit the wrong timing and it just looks super awkward because they just did not get that push from the substrate. Um, but
and it gets
we have another paper coming out that utilizes our virtual reality system
nice.
with a compliant substrate as well. And we were able to recapitulate that the model stays in contact longer because the takeoff powers and velocities are dampened by reducing the quality of the latch.
Interesting. And and there's also like just so in the diver part too, is there some strategy of leaving some force generation capabilities during the recall? So you get max board bend on the way down, but the rotation happens on the way up.
Uh,
And so our model simulations were able to show that, right? So, but it's like strategically determining how to regulate the latch and reorienting the force. So it's
right.
very, this is really wonderful. So keep going.
Thank you. Okay. So I could talk about power amplification for hours, and I did just recently give like a 2.5 hour talk on this topic over a long period. So lots of other systems we could talk about, and I will say my systems, which were the frogs, were probably the least interesting of them all compared to these amazing invertebrate systems and the capacities that they have. Kisa.
So, Manny, I just have a question here,
Sure.
and that is like, I don't know whether you know this pretty cool paper from Walid Farhat and Hugh Hurd that
Yeah.
was published in PLOS back in like 2010. But
Yeah,
I
the
think
two
it
muscle
also,
system.
yeah, I think it also relates to Jill's area.
Yeah.
And so the idea is that, what they found is that a pair of antagonistic muscles operating together that were impedance matched to the load, which seems to really fit into your paradigm,
Yeah.
could produce 7 times as much mechanical power, 7 times as much mechanical power as a single muscle.
Yeah.
So I think the question is like, can you start to incorporate like, antagonistic muscles acting at a single joint into this kind of analysis?
Yeah. Initially when we were working on these topics, I was of the opinion that muscle relaxation rates are simply too slow to really be any good as a latch. I've come around on that to some degree, mainly because my view of systems that utilize elastic energy storage has been greatly broadened. And to me, the idea of chasing the fastest and the most perfect system, I think has actually hindered our understanding of these systems because we've focused on these high performance isolated examples, um, which effectively made me think, no, even the fastest fiber type, that relaxation rate simply takes too long for the force to get out of the way if an antagonist is holding it. But I'm of the opinion now that there are scenarios where bad latches are okay, where in fact, the compliant substrate is a great example where messing up the latch actually was beneficial to the overall performance of the system. So I'm much less constrained in my thinking about what can and can't be a latch. And I have come around to the idea that you can use an antagonist as a latch, particularly if that antagonist not only is deactivating, but that you have the capacity or you have other structural features that allow the moment arm of that muscle to also change rapidly during deactivation. Now you're really amplifying that drop in force, which is what you need to have a good latch. You need that force to get out of the way. Like you need it to be high enough to hold the system, but to get out of the way as fast as possible. I'm starting to come around to the idea that you could play around with muscle moment arms um, and make that deactivation fast enough to act as a latch.
Yeah,
I think also, go ahead.
after you, Jill.
I was gonna say, going back to the two joint muscle parts of this. See, the two joint muscles are really interesting because it controls like 50% of the mass above the hip, if you will. So you can get kind of a 2-for-1 here in controlling center mass position as well as the inertial aspects of it, like to hold it. This again is that resistance part, but this is like why, going back to, I think where you're going, Keith, is looking at the system, right? The combination of multi-joint control and how that effectively regulates these things that you've been able to simplify in your model, which is very exciting, right? So that was all it's going to say, because I think there's something about the system dynamics that we want to bring back in for these latching mechanism, as well as the team of muscles that are involved.
I agree. And I think, in fact, I really feel like this aspect of the field has been hindered by chasing the fastest, the highest accelerations, like the most impressive systems which are fascinating and amazing, but they are divorced from some of the constraints that lots of other systems deal with. And I think of this as being ubiquitous. If you have a tendinous structure in series with the muscle, you simply can't get around it. When the muscle produces force, the tendon will stretch, whether you want it to or not. Like there's no way around it. And so there's lots of interesting complexity that I think we can learn from studying slower, less impressive systems where some cycling of energy is still going through the tendon.
Yeah, and my final point was just going to be that when muscles are changing length, we tend to think of like there's a canonical EC coupling delay. There's a canonical, rate of muscle relaxation, but those are measured under isometric conditions. And as soon as you let the system move, those pretty much go out the window. And so like the length dependence of the rate of force development and the length dependence of the rate of force decay could substantially, sort of, obviate some of the, thinking about really long delays and muscles being bad latches.
Yeah. Great. Okay. Last but not least, I want to talk about power attenuation. I think this is actually an area that really needs our attention. There's only been a handful of folks who focused on this aspect of tendon function. I think it's really cool and has some broad implications. So here's a couple of experiments we did, really simple stuff, like nothing hard. Like this is as simple as it gets. We have an isolated muscle tendon unit right there where we're stimulating. We are driving it through this little stretch, and the gray is when we're stimulating it. Then we just change the rate at which, or the velocity of that stretch that's applied to our muscle. So really, we're just altering the velocity of that stretch. We're trying to actively stretch the muscle, but as you'll see, what we have is a pretty compliant spring in series with our muscle. And so as a result, it's actually pretty challenging to stretch the muscle fibers, okay? So here's a pattern that we get. So we've got length up on top. We have force on the bottom panel. We have length of the muscle tendon unit up on top. That's effectively the length change we're imposing onto the muscle tendon unit. The dark black is where we get the highest velocity for the muscle tendon unit. What we actually see in terms of the behavior of the muscle fiber is that as we activate the muscle fiber, even though the muscle tendon unit is being stretched, our muscle fiber is shortening. It's shortening for much of the stretch phase, and then the tendon is stretching our fascicle. So in this first phase, the muscle is shortening, we're stretching the MTU, so we know the tendon is stretching. And once we get past peak force, the tendon is loading our fascicle. What's most interesting is look at the velocity of stretch applied to the MTU compared to the velocity of stretch applied to the fascicle by the tendon. The fascicle velocity is much lower, which is a good thing, because when we stretch our muscle fascicles at a high rate, they tend to get damaged. When we stretch those muscle fibers to really long lengths, they tend to get damaged. So here what we're seeing is the muscle is shortening, so it's moving it to a safer spot. Then it's being stretched by the tendon at a lower velocity than we were stretching the MTU. So really, the tendon here is acting as a mechanical buffer to limit the power input to our fascicles. We're still dissipating the same amount of energy. We're just doing it in a much safer way by operating at shorter lengths and by operating at lower stretch velocities. Plus, the tendon is a known entity to the muscle, whereas the environment is an unknown and unpredictable entity. So having this material in between our environment and our muscle allows us to load the system in a much more repeatable and dependable way. Here's a summary of that data, and this was work I did with Tom Roberts when I was a postdoc. We have fascicle velocity, and if the fascicle velocity was the same as the MTU stretch we're applying to it, so let's say there's no tendon in place, you would simply follow that dotted line, right? However, the fact that the tendon is slowing down the rate of stretch that's applied to our muscle fiber means that we're getting lower stretch velocities applied to our fascicle, even as our rates of stretch start to get higher. Kisa?
This is really cool, Manny. So I'm just struggling to understand, is the gray bar activation, and is the activation coming on exactly at the beginning of the stretch?
In these experiments, they came on right at the beginning of stretch, yeah.
And how have you looked at how the dynamics of the MTU fascicle interaction change if you change the timing
Yes.
of the activation?
Yeah. I had that in there, but I took that experiment out for time purposes. Long story short, well, first of all, pre-activation, anticipatory activation prior to an impact or an impending eccentric kind of behavior is very, very common. Like, it is the norm. And so we did want to look at, well, what are the benefits of turning that thing on earlier prior to the stretch? So we did experiments where we did it 50 milliseconds before the stretch, right at the onset of stretch, 50 milliseconds after the onset of stretch. And what we found was actually the rates were, the maximum loading rates onto the fascicle were largely the same. What really differed between those was the operating lengths. If you activate early, you get much more shortening. It moves you onto the ascending limb of the force length curve before the stretch happens. So it kind of shoves everything onto safer and shorter lengths by going early, whereas the late activation didn't have that. There was less shortening and then the stretch. And so you were sort of operating at maybe questionable lengths. And we would start those at like 1.25 of L naught, for example, just to be at sort of slightly longer lengths. But yeah, it has an important effect on where you're operating on the force length relationship when you do the activation. There were a couple of other interesting pieces of data. The total energy is the same because we're imparting the same amount of energy into the system every time, and all we're doing is changing the timing. So it's just kind of like the shape of that fascicle length change trajectory is what differs.
But that's really important for risk,
Indeed.
right? Yeah,
Indeed.
so this is a nice part where it's not just energy, but risk aversion, right? So,
Absolutely.
and this is really important in this unsteady movement things that we're trying to all study.
Indeed.
Very cool.
Okay.
Maybe I can just say this, sorry. I mean, this is just a dumb comment, but maybe something that's worth looking into in the future is like from the stuff that I've done modeling, Monica's guinea fowl and James Wakeling and Nikolai Konow's rats, which are bipedal and quadrupedal. It seems like if you're quadrupedal, you have less pre-activation. So you can kind of save that. So you have maybe shorter total duration of activation, and you're saving the energy because if you've got 4 legs, you just don't have you have more bandwidth for.
You're spreading risk across 4 limbs.
Yeah.
Yeah.
And to my knowledge, nobody's really looked at that, but I think it would be an interesting question.
I mean, I'll show some in vivo data on turkeys that we have, but that's a bipedal system, so they pre-activate. Okay, last experiment I want to talk about, same exact thing. The only difference we did was we changed the temperature again, mainly because what we found was that let me go back actually. What we found was the rate of this rate here really depends on the rate of relaxation in the muscle, because that's really what's determining the release of force from the tendon. So if the faster the muscle deactivates, the faster the tendon is going to load the muscle. So deactivation rate ended up being really a critical parameter for determining that maximum velocity. And so we decided to try to shift it by doing these temperature perturbations where we can increase the deactivation rate as the system gets hotter. So here are the patterns we see at 10 degrees and at 30 degrees. What you'll notice is at 30 degrees, because the muscle can develop force much more rapidly, we get this initial shortening right there before the tendon stretches the muscle. Whereas at 10 degrees, because the muscle is being kind of slow, we're not seeing that initial shortening happen. And so in fact, that early shift in the position of where the muscle is operating, that's no longer there. And then more importantly, as we change our properties, what we see is that, okay, that fascicle shortening is much less at 10 degrees early on. But then later on, we find that the fascicle lengthening is a little bit more when we have it at higher temperatures, and the rate at which it's being loaded is significantly higher as well. So the stretch rate applied to our muscle fascicles increases as temperature increases because we're increasing the deactivation rate. And in fact, those two things, so this was the relaxation rate that we quantified in our experiments, really consistent pattern across those two variables. To me, this not only explains, okay, well, what makes sense in terms of protecting the muscles, it also explains an interesting pattern in the literature where some folks have shown that fast glycolytic fiber types tend to have more propensity for eccentric muscle damage than slow oxidative fibers. That to me is consistent with the idea that if the tendon is loading the muscle, that rate really depends on the muscle's relaxation rate. And if you increase that relaxation rate, you're increasing the rate at which the tendon loads the muscle. That make sense there?
Say that one more time. So the relaxation rate.
So as you increase the rate of relaxation, that drop in force is really is equivalent to the recoil of the tendon, right? So then you're recoiling, you're allowing the tendon to recoil faster if your muscle is relaxing faster. Okay. Last but not least, here's in vivo data of that same phenomena where we have a turkey that's dropping from the ceiling. We took it up about 2 meters high, maybe it was 1.5 meters in a harness, no wings, wings are clipped, so it can't really slow itself down. It's a direct drop onto its hind limbs. It's a bipedal animal. And what we find is, you see that pre-activation right there, that's in the aerial phase. So the muscle's turning on well in advance of impact. Once impact occurs, we have that rapid rise in force. However, at impact, when we have a rapid rise in force and an ankle that's flexing, and we're measuring activity from the extensors, we see our muscle fibers shortening like we did in our isolated muscle preparation. So in this case, our fascicle lengths are shortening, even though our ankle angle is flexing, which would tend to stretch our muscle fibers. So this is really energy going into the tendon at impact. Whereas later on, as the muscle begins, as we get to the sort of force decay phase here, we see that that's when our muscle fascicle gets stretched. So during force rise, we're not stretching the muscle is shortening. But during forced decay or relaxation, that's when we start to see the stretch. We would argue that that is a much slower stretch applied to the muscle fascicles than would otherwise be occurring without having that tendon in the middle. You can sort of see that the MTU power is shown here in black, that input energy into the MTU is 600 watts per kilogram, but the input power into our muscle fascicles is more like 200 watts per kilogram. So we're really buffering the mechanical power input. We're attenuating that power that's being applied directly to our muscles. And so this schematic simply shows that as we're coming down, when we land, the ankle flexes, it stretches that tendon while the muscle is actually shortening. Then the tendon recoils, allowing imparting that stretch onto the muscle in a much more predictable and more and safer way. Okay, I've talked for an hour 20. Let's see what Kisa has to say.
Sorry. I think this is all just so cool and I'm really excited about it. But I was just going to say, I think it would be really, really interesting in this particular experiment to see how much of like the MTU force is due to like the viscosity of the tendon in addition to the elastic force. So like one of my little pet peeves is that, we do these stretches and we measure the force and we tend to assume that everything is just a spring. But I think like this is a case where the time constants and the viscoelastic behavior could be super, super important in determining the dynamics.
I don't disagree. I think that's absolutely true. Certainly, at the very least, 10% of that energy is going to be dissipated by the tendon itself because it's got hysteresis, probably more like 20. Plus, I don't think we've really studied the behaviors under faster, more sort of physiological rates to see if that hysteresis is what the number is in terms of once you start to really push the rate of loading up some. So I agree, and the stiffness of the system could change as a function of the loading velocity as well. That's probably something we could look at, because in this experiment we did have drops of, I think, 0.5 meter, 1 meter, and 1.5 meter height drops for the bird. And so the loading rates are going to differ to some degree, and we could potentially back out some of the tendon properties and compare them across those conditions.
Are you able to look at what's happening above just the ankle? Because that, like things in human landings, the ankle will bottom out at some point. You'll
Yeah.
hit max dorsiflexion, but this is where the other rest of the system comes in play.
We can, but we didn't. Is
Is it hard to? Yeah, I imagine it just adds layers of complexity
well,
here, but it's
the
interesting
knee
to
is
see the
kind of
team.
annoying in birds because it's like hidden.
Ah,
So in terms of joint dynamics, you kind of lose it in the body of the bird. So you're seeing where the knee is. It's up here. So in some of those, it gets a little bit obscured by the rest of the body, but it's doable. Particularly like the body compresses when
yeah.
it lands, and so that's when it becomes obscure. That's why so many people study the ankle of birds, because it's so nicely
It
visible,
gets-
and the knee kind of gets shafted. Admittedly, this is a biarticular muscle, and so the actions of at the knee are also influencing the behavior of the system.
Because I think that plays into Kisa's question, because it's like it's not all responsible. The ankle joint is not totally responsible for everything, right? So, but I think this is also a great time to ask what's enough. It's like maybe we can't see the knee and we're not doing joint kinetics or whatever, but what we really want to know is how the team of muscles might work to deal with that inertial measure regulation, right? So there may be ways to get insight without having to make it too difficult. I don't know, just curious.
No, it's
Yeah.
doable, and I think the better way to do it is with an X-ray system, with a high-speed X-ray system, where you can visualize the skeletal elements much better, regardless of whether they're obscured externally or not.
Could you also do it like with sono instead of just the ankle oh, I guess you have the fascicle length. So it
Yeah.
seems like you could back out the knee angle change from the fascicle length and the ankle angle.
You'd have to make some assumptions about tendon stiffness, but yeah.
But you know, segment lengths, right? And
Segment length we know.
deformation of the whole
Yep.
system. So you could,
Yeah.
you know, things are all connected there. So it's a geometry
No,
problem.
in fact, we had center of mass sits right above the hip. And so we know exactly where our center of mass markers are relative to the hip. And so we could back out exactly actually where the joints are.
And it seems like that on the, you know, sort of in the, well, sort of after 100 milliseconds when the black and the red lines are deviating, that like in direction particularly, that's going to be the effect of the knee angle, which seems like it's big.
Yeah, I agree.
And from a control perspective, I really enjoy the fact that there's recognition that the control might be different earlier rather than later, which is right.
Yeah.
This gets missed all the time. And so I really appreciate this.
Well, it's, I think in terms of sensory motor information, I think this suggests that particularly the 1B afferents aren't really to be trusted, aren't really getting directly relevant information in a system with a lot of compliance present in series, simply because the MTU is stretching and the fascicle is shortening. And so it's really going to attenuate the proprioceptive signals that are being sent to the central nervous system. So I think an interesting way to sort of think about this is, does Golgi information now become much more important in these systems compared to stretch receptors, where we've largely decoupled the behavior of the fascicle from the external mechanics of the system? So to me, I think there's something really interesting about hierarchy of information in systems where you've got a spring kind of in the middle.
And this is the part about practice in a habitat, right? So because it's a predictive thing, right? You got to turn on before contact. You kind of have to know what you're encountering. And then that plays into some of the foot position relative to center mass trajectory. And are you in the ranges of step lengths and rates that you prefer, or now have we pushed you beyond? So this is the part, some of the Summer Institute stuff that Craig McGowan's group and Clint's been doing in the field will feed nicely into the conversation.
Absolutely.
Great.
Yeah, I think this part really brought a couple of things to my mind when I put it together. One was that the limits of stretch receptors and the information they send, but also if you're not able to stretch the fascicles at any reasonable velocity, it also has implications for things like residual force enhancement and passive force enhancement if you're sort of buffering the fascicles from experiencing much stretch in vivo. So it's a system that's buffered by sort of a viscoelastic spring. Okay, I think that's all I had. I just wanted to rehash where we've been. We can conserve power, we can amplify it, or we can attenuate it utilizing in-series elastic elements, which really should be thought of as viscoelastic. Okay, we have 2 minutes left. I think maybe we can give that back to folks, but I will put the feedback, the workshop feedback QR code up for people who need it.
Great. Thank you so much. This was a great presentation. I know I learned a lot, so I appreciate it.
Thank you, Victoria.
Thank you, Manny.
Thank you, guys.
All right, bye everyone.
All right, bye-bye.